Hey, how's it going? This is Daniel Gomez in the Snyder Lab in the Department of Genetics, Stanford University School of Medicine. I wanted to go over the long non-coding RNA landscape of endurance training, exercise training, as well as long non-coding RNA mechanisms. So the link are long non-coding RNA structure function relationships are genes that specify long non-coding RNAs and occupy a large fraction of genomes of complex organisms. Link RNAs are generally range from a size of around 1 kb to longer than 100 kb and have a modular structure. They're often multi-exonic and highly alternatively spliced, meaning they can have multiple exons and spliced in different ways. And there's a feature that's not obvious before the advent of this is a feature that was not obvious before the advent of high-depth sequencing. So they contain a higher proportion of GC-AG splice sites and therefore are less efficiently spliced than protein coding transcripts, which are properties associated with alternative splicing, or what's known as AS. So alternative splicing has unsurprisingly been shown to alter the function of link RNAs. Many link RNAs are associated with chromatin modifying complexes, and they are transcribed from enhancers and the nucleate phase separation of nuclear condensates and domains, and indicating an intimate link between link expression and the spatial control of gene expression during development. Link RNAs also have important roles in the cytoplasm and beyond, including the regulation and translation, metabolism, and signaling. Okay, so I wanted to show a figure here of alternative splicing of link RNAs, where we can see the exon at the end, intron, and then exon at these red lines, vertical lines, and then we can see how it splices in different ways through the dotted lines. So and this is an example of link RNA in chromosome 21, where we have mostly alternative splicing and only a sliver of constitutive splicing. And next is here we have a domain of repeated architecture of link RNAs. So we have domains and repeated domains. Domain 1, domain 2 is repeat domains, and then a poly A tail and a 5' cap. So I'm from 5' cap to domain 1, repeated domain, and domain 2, and then poly A tail, like a normal mRNA, but it's a link RNA. And so the different functions are DNA binding, with gene regulation. So DNA and Apollo, that is an Apollo DNA. Protein binding PNCTR, PTBP1, YYUCUY, gene splicing, and protein binding with SPEN and PRC2. So a repeat and exist um, uh, gene, which is or transcript that's bound by the protein and then epigenetically silenced by these two proteins, SPEN and PRC2. And then we have RNA binding with a Staufen one. It binds to the mRNA and the TINCR for mRNA stability. So several points here. We have targeted RNA sequencing that's revealed as human chromosome 21 and pervasively transcribed into long non-coding RNAs that are link RNA exons, and they're almost universally, but not randomly, alternatively spliced to form diverse and complex isoforms. So these structures are also, domains are also modular. So they have link RNAs that fulfill a range of functions that include targeting DNA, such as in the case of this Apollo promoter loop, binding to the other RNAs. For example, we have terminal differentiation induced non-coding RNA for the RNA binding that I showed, TINCR, and potentially involving RNA binding proteins such as thauphin-1. And the recruitment for proteins, for example, are pyrimidine-rich non-coding transcript, PNCTR, which is pyrimidine-rich, and then pyrimidine track binding protein is recruited, BP1, we can see over here. And that happens through special RNA motifs. And X inactive specific transcripts exist, recruiting splicins, homolog, SPIN, or SPIN. And the polycomb repressive complex, PERS2, PRC2, is a polycomb repressive complex that acts. It's a bunch of them. So there's a bunch of PERSes that are a polycomb represses the DNA, as well as exists an X inactive specific transcript, which is a well-known gene. Perhaps in concert, we have a subject of active exploration and debate. Whether they work together or not, we don't know. You have to debate that and explore. Okay, so modular functional domains can be repeated with a link RNAs or in multiple different link RNAs. We can understand that for every repeat here in the domain, you have multiple different link RNAs. And that's what makes it modular function. So these are the structure and function of link RNA relationships. So the link RNA endurance exercise training, and we have here a MoTrPAC. The rat F344 Fisher 344 rat underwent 8 weeks of training for males and females, where we have 8 weeks of training, and then 18 different organs that were either sedentary, 1 week, 2 week, 4 week, and 8 week. We can see all the different organs, and their link RNAs were measured. So we can see link RNA exercise molecular landscape for multiomics, 18 tissues, 4 time points, that were sex specific, and we get the this landscape of long non-coding RNAs and endurance exercise training. So the transcriptional landscape of these link RNAs are in skeletal muscle mainly. What we'll look at is after 8 weeks of aerobic training. So specific for that cluster of skeletal muscle and 8 weeks of aerobic exercise. All right, so we have tissue-specific associations between the link RNAs that were chromatin accessibly at regulatory region. And this is important because if you want to look at the link RNAs, then we have to look at the chromatin accessibility sites at these regulatory regions. Next, we'll go over the link RNA EET highlights. Okay, so endurance training altered 759 unique link RNAs across 18 tissues. The link RNA profiles were largely tissue specific, being the adrenal, hypothalamus, and brown adipose tissue, or BAT. And those were really pronounced sex specific differences dominated in the link RNAs landscapes. And they integrated with chromatin accessibility data revealed regulatory associations. And these gene link RNA correlations in adipose tissues are associated with immune regulation. So this is what we're going to go over. Okay, so up top we see the schematic, which I went over, and then below it we see the total number of link RNAs expressed detected in each tissue after filtering for a mean FPKM greater than 1, and that represents a reliable expressed transcript, including downstream analysis. So we can see here all 18 organs, and then the number of DELs, or differentially expressed link RNAs, DEL in NCS, and then the weeks of training, week 1, week 2, week 4, and week 8, and then the percentages, as well as the Z-score. So that's what we can see in C, D, and E, and F in this figure are all the different expressions in 18 tissues, and then the the number of DELs, the weeks of training, the percentage, and the z-score. You can see that in week 1 through 4, in the thalamus, hypothalamus, have they're in the 30 range, and then the brown adipose tissue, we see some in 29, 23, and 32. And then in the adrenals, we have 28, 25, and 34. And then the white adipose tissue, we can see 38, 26, 30, and 23, which are all the different weeks and how much weeks of training that they have for each tissue. And that's expressed. So that's the temporal distribution of the DELs across exercise training time points. So that goes over everything for that map. And then E and F are just sex-specific distribution across the tissues, showing the proportions of the link RNAs differentiated regulated males that are in blue and females that are in pink animals. And those distributions are normalized for link RNA expression changes across tissues represented as z-scores. And those illustrate the overall magnitude and the variability of link RNA regulation during endurance training. Next, we'll go over these components for male and female. So we did a t-SNE projection of global link RNA expression profiles across tissues in female and male rats. Each point represented a tissue-specific sample with four points. Tissues per tissue corresponding to 4 weeks of training time points. So 1, 2, 4, and 8 weeks. Samples are colored by tissue. So the clustering is primarily driven by tissues type rather than training duration, indicating a strong tissue-specific link RNA transcriptional program. So we can see that in these clusters here in male and female. So in A and B, we can see adrenals in male, and then brown adipose in male, cortex, um, skeletal muscle vastus lateralis in green, as well as skeletal muscle gastrocnemius in darker green, and then kidney and liver in purple, light and dark purple. And then in yellow, we have the white adipose tissue as well as the heart. And the same thing in the female, but it's more condensed or closely together. So the males are kind of spread out and the females are together in this T stochastic neighborhood embedding projection. And then in C and D, we have Pearson correlation heat maps showing the similarity of link RNA expression profiles across tissues and training time points in female and male rats. So we have females and males, and this is a Pearson correlation. So we have by weeks, the Pearsons by weeks. And it's for each organ. So each organ has a color. So there's 18 different colors in this Pearson projection. And then it goes up for females and then goes up for males. And this heat map where we can show the similarity of link RNA expression profiles across tissues and time points in female and male rats. Okay, so next we'll go over the percentage of differentially expressed link RNAs, or these DELs, these DEL NCSs, and DELNICs maybe. So we have with or without annotated human orthologs across tissues, and we have rat-human ortholog, which was determined using annotations from the rat genome database, where we see the blue bars indicate that these DELNICs were lacking annotated human orthologs, whereas the red bars indicate the transcripts. So the transcripts with predicted rat-human orthology. And then the upset plot that shows the number of shared DELs. The number of shared DELs across 8 internal organs. So they only did 8 internal organs here. The heart, colon, lung, liver, spleen, and kidney, and small intestine, as well as adrenal glands. The bars indicate the number of DELNICs shared among the tissue combination shown here on the left. So we can see the 8 different organs and the number of shared DELs or delnics. And then we can see chromosome 10, chromosome 7 coordinates. So we have KICKMB4 as well as alternative splice 2 that we can see alternative splicing here reading from the left and then to the right, as well as the ensemble gene in chromosome 7 reading to the right. And then they're focusing here on the last one in the heart, kidney, and small intestine and adrenal gland. So the last three are kidney, spleen, and adrenal, as well as the heart in the last one. So that's for the kicking and B4 and kicking and B4 OS2 or OS2. And those are it's shown as the reason why they did that is it's conserved. So it's conserved in the rat and mouse genomic context. So that's more a good translational study where we can do a multi-organism, compare the two, and show that it's conserved in this genomic loci representative delnic. All right, so here we have the transcriptional landscape of lincRNAs in skeletal muscles after 8 weeks of aerobic exercise for principal component analysis PCA of the global lincRNA expression profile and showing the separation between gastrocnemius and the vastus lateralis across the exercise training time course. So we have the tissue in dark green, the gastrocnemius, and light green, the vastus lateralis. And then we have these red and blue dots for male and female. And then the skeletal muscle vastus lateralis and gastrocnemius here on the right, you can see the mean Z-score and weeks of training for males and females. We have a mean Z-score distribution of expressed lincRNAs, delnics, and SKMVL and SKMGN across the training period. And there's data showing the separation in male that's in blue and female that's in pink. So the female in pink uh, is under the male in the gastrocnemius, and then above in the vastus lateralis, which are two different muscle organs. Okay, so the Venn diagram shows an overlap of del nits between the two. So we can see 45 and in the vastus lateralis and 16 in the gastrocnemius, and an overlap of 2 between, highlighting that most of the lincRNAs are unique to each tissue. So temporal expression patterns are shown in D with the log fold change expression, where we can see all these ensemble genes, and then going from sedentary for each cluster, sedentary 1 week, 2 week, 4 week, 8 week, in vastus lateralis, and the gastrocnemius. So those are the temporal expression patterns of selected delnics across the training time course in skeletal vastus lateralis and skeletal muscle gastrocnemius. Okay, so the next figure here, a comparison of transcript length log2 nucleotides, where we've seen delnics and differentially expressed protein coding genes, or the DEGs, or DEGs, in skeletal muscle VL and GN. So you see that it looks like a violin plot. We can see lincRNAs, protein coding, lincRNAs, and protein coding for each vastus lateralis and GN on gastrocnemius, and a comparison of the exonic GC content percentage between delnics and DEGs in skeletal muscle VL and skeletal muscle GN. So that's how much GC, guanine, and cytosines are in each exon, and they compared the percentages of those for the delnics and the DEGs and the DEGs. In G, we have distribution of predicted minimum free energy, MFE, for del-nix and degs, and each tissue showing no significant differences in RNA secondary structure stability. Coding potential analysis for del-nix in skeletal muscle, vastus lateralis, and skeletal muscle GN, and is shown in H and I, where you see the coding probability CPC2 and coding probability RNA samba. Okay, so yeah, those two predictions. So those two predictions are measured across each other. And then in J, here we have a genomic context and cross-species sequence alignment, identifying the del-nic ensemble gene corresponding to apelin. So the apelin locus shows a partial conservation across species. So we have here in J, you can see 77 amino acids, and that's that shows we have a cross-species alignment right under that, where we can see Rattus norvegicus, Mus musculus, Bos taurus, Homo sapiens, Macaca mulatta, all aligned, all those species aligned, identifying the del-nic to be apelin, which its genomic coordinates are there. In chromosome X, 127 million or so down. And then in K, we can see a predicted three-dimensional structure of apelin wrap protein in human. Green, it's you see it in rat, and in gray, you see it in human. So the models correspond to the AlphaFold predictions retrieved from AlphaFold protein structure database. And the UniProt codes are accompanied that. You can see the UniProt codes over here. The structure, I mean, it looks really good. You can see that there is a difference between the two species, but probably it's the same length that makes a different conformation. And then the temporal expression profile in the apelin locus during an endurance training is shown. And the blue lines represent the male rats, pink lines represent the female rats. And we can see that over weeks of training, from sedentary to 1 to 8 weeks. All right. And then getting into the component analysis, we have PCA of a global lncRNA expression profile showing the separation between brown adipose and the white adipose. Samples across the exercise training time course. We can see both sexes, brown adipose and white adipose. And then mean z-score distribution of differentially expressed lncRNAs in brown adipose and white adipose. A Venn diagram illustrating the overlap of the del-nix between the bat and wat adipose tissue, showing mostly the lncRNAs that are regulated in tissue-specific manner. So we see those, the expression for each gene or lncRNAs there, and then the temporal expression patterns of selected del-nix across the training time course in both tissues are shown in D, and the E has the expression profile, and distinct for male, where it goes up and down in a distinct pattern across the exercise time points. And then in comparison of the transcript length by nucleotides, we see expression of del-nix and differentially expressed protein genes and brown adipose and white adipose, compared to in the comparison of the exonic GC content between del-nix and DEGs in brown adipose and white adipose tissue. So the distribution of predicted medium free energy, or the MFE values, were computed for Dell-Nix and DEGs in brown adipose and white adipose tissue, indicating that the significant differences in RNA secondary structure stability. So this is one way to measure RNA secondary structure stability, where the RNA begins to take form. We can measure the medium or minimum free energy predicted values, and the distribution of that for each organ. So the coding potential analysis of link RNAs in brown adipose and white adipose are based on the CPC2 and RNA Samba predictions. And we can see those here in blue and red and yellow and red too. And then we have gene ontology enrichment analysis derived from Dell-Nix and DEG correlation networks in brown adipose, shown here in K, the white adipose. And then highlighting the pathways with immune regulation, we see immune regulation and inflammatory signaling. So in blue, we can see about 100 genes regulating the signal transduction and cell communication. And then we have the regulation of B cell activation, probably 12.5 genes, and 15 genes in a cellular response to biotic stimulus. Here are some scatter plots with the density contours, and this shows the relationship between link RNA expression and ATAC peak accessibility as a function for genomic distance between the link RNA loci and nearby ATAC peaks. So the x-axis has the distance to TSS, which is the transcription start site. And the y-axis is the Pearson correlation between the link RNA expression and the ATAC peak accessibility across screening time points. So that's what we have for each organ, scatter plot of a density contour. So you can see the skeletal muscle, gastrocnemius, the lung, the brown adipose, the kidney, the heart, the white adipose, and the liver. And then we have activity. So activity scores analysis in the liver, integrating correlation between link RNA expression and ATAC peak accessibility and genomic distance between peak and the TSS of nearby protein coding genes. So we can see here PDE8A, TBXA2R, TMX1, impact, AP3B1, ZFYVE9, that all have activity scores, as well as SQLE or SQLE in the liver. So this is each point. Each point represents a link RNA and ATAC peak gene association. And then the gene ontology enrichment analysis of protein coding genes identified in positively correlated link RNA ATAC peak gene triads. And we have a sterol biosynthesis, a cholesterol biosynthesis, subtelemic heterochromatin formation, and regulation of endothelial. So these are the ones that go up, are the sterol biosynthesis process, the cholesterol biosynthesis process, and then the subtelomeric heterochromatin formation that goes up, as well as the cholesterol metabolic process. There's also some acetyl-CoA metabolic process, and other goes as well. Downregulated is regulation of endothelial cell migration and G-coupled protein dopamine signaling pathway that's downregulated, and respiratory chain complex 4 assembly, which is right before the ATP synthase, and then and the mitochondrial cytochrome C oxidase assembly. So those are the enrichment pathways that are separated for upregulated and downregulated gene sets. So these RNA landscapes in endurance exercise training was leveraged and enhanced with comprehensive multi-omics datasets generated by MoTrPAC to gain deeper understanding of adaptations induced by progressive endurance training. Okay, so lincRNA expression is not only inherently sex biased, but also dynamically modulated in response to physiological challenges such as exercise. So the tissues directly involved exercise, such as the SKM and the heart, were expected to exhibit a higher abundance of differentially expressed lincRNAs. And the greatest regulation of the lincRNAs were hypothalamus adipose tissues, brown adipose tissue, and white adipose, and the adrenal glands. The hypothalamus is the primary tissue responsible for control of the whole body homeostasis. Adipose tissue and adipose tissues are critical for the regulation of whole body metabolism, as well as maintaining homeostasis and the response to various stimuli. These changes involve regulation of fatty acids, energy and lipid metabolism, hormonal and enzymatic activity, expression and secretion of adipocytes, which apelin and adiponectin, and immune and angiogenic and fibrotic remodeling. And these are all shown by MoTrPAC Study Group, as well as other studies. So besides this, the beneficial exercise or the beneficial effects of physical exercise on the modulation of adipose tissue are well known, highlighting both positive changes in immune and inflammatory responses, as well as lipid metabolism. And Wu et al. Shown that in 8 weeks of endurance exercise inhibited the expression of the lincRNAs in SRA in obese mice. In an SRA, this inhibition could trigger the activation of P38 and J and K pathway, suppressing the expression of P-PAR gamma and its downstream genes, resulting in the improved lipid metabolism. So analyzing different molecular layers affected by exercise is important. The differential expression and regulatory role of lincRNAs at various levels are DNA, RNA, protein, and we anticipate that these findings will enable hypothesis-driven exploration of lincRNAs in response to exercise and training. The identification of lincRNA regulation across multiple tissues in response to endurance training, demonstrating the complexity of exercise-induced molecular adaptations, and establishes a foundation for understanding the regulation by non-coding RNA in a physiological context. Thank you for your time.